Hi dear students, good morning. I'm Dr. Mashur. Welcome to my class. Today we can discuss DNA sequencing. Okay, we can discuss DNA sequencing. Okay, in this we'll discuss what are the basic steps, what are the basic methods involved in DNA sequencing, and what are the modern techniques which is used in DNA sequencing. Let's see DNA sequencing. Okay, DNA sequencing. The DNA sequencing is the process of determining the sequence of nucleotide bases present in a piece of DNA. Okay, DNA sequencing is the process of determining the sequence, nucleotide sequence present in a DNA. Okay, that means nucleotides are the building blocks of DNA. Okay, nucleotides are the building block of DNA, deoxyribonucleic acids, and that contain four bases like adenine, thymine, guanine, and cytosine. And this sequence of this nucleotides that's code for the different proteins and others RNA. Okay, this nucleotide sequence that code for different RNA and proteins. Okay, and the change in this nucleotide sequence that will change the structure of protein and the function of proteins. Okay, so that DNA sequence, that means arrangement of nucleotides in a DNA, is that much important. So the DNA sequencing is the process of determining the sequence of nucleotide bases present in a DNA. That means the DNA sequencing determine the arrangements of nucleotides bases in a DNA. So, and it's having DNA sequencing having a wide application in various fields like in agriculture field, medical field. So many fields are we having a wide applications. So to determine the sequence of a gene and to determine the mutation, so that we can use this DNA sequencing in various fields. And DNA sequencing can be done by different methods. Okay, DNA sequencing can be done by different methods. Like we can see the basic methods like MAXAM-GILBERT method and chain termination method. These are the basic method. Okay, MAXAM-GILBERT method and chain termination method, and some other methods also like automated sequencing. Those are the modern methods: automated sequencing, pyro sequencing, NGS, next generation sequencing. Okay, next generation sequencing. Okay, in this video, I would like to explain MAXAM-GILBERT method of sequencing and the chain termination method of sequencing. And remaining the new generation, the modern technology like automated sequencing, pyro sequencing, and next generation sequencing, I will explain in the next class. So first we can see the MAXAM-GILBERT model of DNA sequencing. First we can see the MAXAM-GILBERT method of sequencing. Okay, this method of sequencing was developed by Alan Maxam and Walter Gilbert in 1976. Okay, and this method is also known as chemical method of DNA sequencing. Okay, the Maxam-Gilbert method of sequencing was developed by Alan Maxam and Walter Gilbert in 1976, and this method also is known as chemical method of DNA sequencing. Without In this procedure, this method of DNA sequencing, chemicals are used to break the DNA. Okay, and this was the method first widely adopted for DNA sequencing. Okay, this method was first widely adopted method for DNA sequencing. Okay, now we can see the procedures, steps involved in Maxam-Gilbert sequencing. Okay, we can see the steps involved in Maxam-Gilbert sequencing. Okay, first we have to take the double stranded DNA that to be sequenced and that is to denature into single stranded DNA. Okay, first we have to take the double stranded DNA that to be sequenced and that's to be denatured into single stranded DNA. Okay, and that single stranded DNA is one end to be labeled with radioisotope. Okay, that single strand DNA 1 and should be labeled with the radioisotope. Normally, P32 is used for a for radio labeling. Okay, and then this labeled DNA samples added into 4 tubes. Okay, this labeled DNA will be added into 4 tubes, and this each samples were subject to chemical treatment that specifically destroy one or two of the four nucleotide bases in of each of four reactions. Okay, and this reaction digests the labeled DNA into different fragments with the varying length. Okay, this chemicals that will destroy one or two of the four nucleotide base in each of four reactions. Sometimes this reaction will cleave G, and in another reaction that will cleave A and G, and in another reaction that will cleave C nucleotide, and the other tube is cleaves C and T. Okay, this figure explain how we will do the Maxam-Gilbert sequencing. Okay, how we'll do the Maxam-Gilbert model of sequencing. Okay, we already told that we'll isolate the DNA and we'll make it into single stranded DNA, and we'll label that DNA. Okay, we'll label that single stranded DNA, and that labeled single stranded DNA will distribute into 4 tubes. Okay, that will distribute this for this radio labeled single strand DNA into 4 tubes, and in that 4 tubes the specific reaction will be take place. Okay, the specific reaction will be take place in that tubes. Radial doubled single stranded DNA. Okay, radial doubled single stranded DNA. We have split into 4 tubes. Okay, 4 tubes. 1, 2, 3, 4. We split into 4 tubes. In this tube, this reaction will cleave A and G nucleotides. Okay, this reaction will cleave A and G nucleotides. And in the second tube, that will cleave the G nucleotide from the chain of DNA. And here this is the third tube that will cleave. This reaction will lead to cleave C nucleotide from the chain, and in the fourth tube that will cleave C and T nucleotides from the chain. Okay, C and T nucleotides from the chain. Okay, after the chemical treatment of these 4 tubes, we'll load this samples in a polyacrylamide gel. Okay, we load this fragment, these samples in a polyacrylamide gel. Okay, after the reaction, we load this 4 tubes in the polyacrylamide gel to separate the different DNA fragments. Okay, to separate the DNA fragment, and expose this polyacrylamide gel reference into for autoradiography. Okay, for autoradiography, because this DNA is labeled, radio labeled. That's why it's give the autoradiography. So after the polyacrylamide gel reference, expose this gel into the for autoradiography, and we can see different bands in the photography films. Okay, we can see the different bands in the photography film. From that, we can find out which are the DNA sequence. Okay, now we can see this. If you come here, this is the downstream part of it. Okay, this is the lowest. Okay, this is the down part of this gel. Okay, this is the uh, lowest part of this gel. Okay, lowest part of the gel. This fragment will be very this fragments are will be small. Okay, this fragment will be very small. Okay, here you can see, you can identify which are this sequence. Okay, see in which well this bands are observed. Okay, in which well these bands are observed. You see in the C plus T. Okay, C plus T reaction, here one band is there. C plus T reaction, here one band is there. and so we can say that this band is a C or T. Okay, C or T. Then we have to confirm whether it is C or T. So just check the well of C reaction. C reaction. Here you can see the reaction. So it's confirmed that it's in C, and C at C plus T. To we have given the band. That's why it's called as a is a C is a C nucleotide. Okay, the C nucleotide. Okay, C nucleotide. Here you can see, for example, here you can see this is the labeled in labeling. Or here one nucleotide is there, that is labeled, and this is the first band attached, first nucleotide attached with this G, and this is C. This is the first one, this is the second one, third one, fourth one, fifth one, sixth one, seventh one. Okay, and the labeling is done with the G nucleotide. Okay, so here this is C because of this is a low molecular weight. Low molecular weight means this is a small fragment of DNA we observed in during the chemical reaction. So that breakage will be here. Okay, after first nucleotide. Okay, sorry, from here this G before the first nucleotide, that breakage will be happen here. That's G and C. This breakage is okay here. So you can see the first nucleotide is a C. And next we can see here any band is there. So next near top of this this band we can see any band is there. That's one band is there. Here it is seen in the C plus T tube. Okay, C plus T tube is there. Now we have to check whether is any band is in the same length, same pattern in the C tube. But here there is no band is there, so we can confirm it is a T. Okay, T. So that T is came because of, you can see after this one breakage occur, and see this between this breakage is occur. That's why is given somewhat high molecular weight. And you have to check any other band is on top of that, and here you can here see here one band. This band is a in a plus G tube. Okay, this band is in a plus G reaction tube. So that time we have to check whether any band is observed in the G. Okay, so here there is no band in G, so we can say this is a. Okay, this is a as A. So okay, this we can say it is as an A. So this break occur here. Okay, break occur here 2 and 3 here break is occur. And next we have to we have to see, and any other band is the same. You can see here in this one band in the C plus T, and one band in C. Okay, one band in C plus T and one band. That's why this may be C or T. Okay, that's why you have to confirm whether this C, that we have to check in the C tube that C is there. Okay, C is there, and so that C breakage is okay here. The breakage is okay here, and after that we have to check any other bands, so that we can see here one band, the G tube and A plus G2 tube also is the band is there. So we have to confirm whether it is A or G. So that time we will check in the G tube, that time G that shown the band. Okay, and after that here also you can see that here back cutting will uh, be happen here, and after that we'll go to that here band is there. Not only C plus T it is there, C plus T is there, but there is no banding in C, so it is a T. And after that we'll go to here in seventh, the here one A plus C to be having one band. So but there is no band in G, so it is a A. Okay, so we can see this is C here, T A C G A A. Okay, this is a sequence. Okay, this is the sequence. So from here we can read that sequence is a C T A C G T A. This is the sequence. Okay, and this is about MAXAM-GILBERT mode of DNA sequencing. I hope you understand this. Okay, I hope you understand this. Now we can see the Sanger's method of sequencing. Okay, we can see the Sanger method of sequencing. Okay, this Sanger sequencing is also known as enzymatic method of sequencing. Okay, this Sanger sequencing is also known as the enzymatic method of sequencing, and this method was developed by Frederick Sanger and his colleagues in 1977. Okay, this method was developed by Frederick Sanger and his colleagues in 1977. And this method of sequencing, that means Sanger's method of sequencing, is commonly referred as chain termination method or dideoxynucleotide method. Okay, this Sanger's method of sequencing is commonly referred as chain termination method or dideoxynucleotide method. Okay, we know that dideoxynucleotide are the modified nucleotides that lack hydroxyl group and on the both 2' and 3' carbon of the sugar of nucleotide. Okay, that DDNPs means dideoxynucleotide are the modified nucleotides that lack hydroxyl group on both 2' and 3' carbon of the sugar. Okay, 2' and hydroxyl group in the case of DDNP. Okay, here you can see this is the DNTP. Okay, DNP, that means deoxynucleotide. Okay, this deoxynucleotide is present in the normal DNA. Okay, deoxynucleotide is present in the normal DNA. So you can see here in the second position is hydroxyl group is removed in the case of deoxynucleotide, and but in the third carbon, the third carbon, there's a hydroxyl group is there in the deoxynucleotide, but in the second position that hydroxyl group is removed because or is removed in this deoxynucleotide. Okay, and we told that dideoxynucleotide is a modified nucleotide that lack hydroxyl group on both second carbon and third carbon. Okay, second carbon and third carbon. You see here, there's a second position and there's a third position. What is the problem with that? If there is no hydroxyl group in the third position means, okay, there is no hydroxyl group in the third position means when we are adding nucleotides into the in the PCR reaction mixture, that time when this dideoxy is attached into the chain of DNA. Okay, when this dideoxynucleotide is attached into the growing chain of DNA, that time this 3' OH is not present in this dideoxynucleotide. Okay, dideoxynucleotide. That's why the next nucleotide cannot able to join with this preceding nucleotide because the next nucleotide will be added into this nucleotide by the OH group of this 3' OH group of this nucleotide, because in this dideoxynucleotide there is no OH group in the third position, so it cannot able to make phosphodiester bond with the coming nucleotide. Okay, coming nucleotide cannot able to make phosphodiester bond. That's why that will terminate the chain. Okay, that will terminate the chain. Here you can see this is our, for example, this is our template DNA. Okay, this is our template DNA and this is the primer. Okay, this is the primer. Okay, this is the primer, and we'll add this when we add this normal deoxynucleotide and a DDNP mixer, that when after this into this primer, when this deoxy dideoxynucleotide, this abnormal nucleotide dideoxynucleotide is attached to this primer, that's that time is this dideoxy does not have any 3' OH group, so the coming nucleotide cannot able to join with this. So the chain is terminated. So this chain is terminated. Okay, this is a chain termination. Chain is terminated. That's why it's called as a chain termination PCR. Okay, chain termination PCR. Here you can see this is another one case. Here this is the up to T is the primer, and in the then in the system that added normal guanine nucleotide, normal guanine nucleotide. So it having a 3' OH, 3' OH, but the polymerase and in the next the adding of the next nucleotide that add abnormal adenine nucleotide, abnormal adenine nucleotide. So Here there is no OH group, so the no OH group, so the polymerase cannot able to join this next nucleotide into that. So this chain also terminated. So this type of PCR is called as a chain termination PCR. Okay, chain termination PCR. Okay, this is called as a chain termination PCR because this chain will be stopped. Okay, this chain will be stopped. That's why this sequencing, because this sequencing need this chain termination PCR. That's why this sequencing also, that's why it's also called as a chain termination method of sequencing. A chain termination method of sequencing. Now we can see what are the steps involved in chain termination sequencing or Sanger sequencing. Okay. Now we can see what are the methods, steps involved in the Sanger's method of sequencing. Okay. Normally, like in the MAXAM-GILBERT method, this sequencing reaction also take place in four tubes, and this four tubes contain template DNA, DNTPs, that means normal nucleotides, and contain polymerase, DNA polymerase, and labeled primer, okay, radial labeled primer, and any one type of DNTPs. Okay, any one type of DNTPs. Any one type of DNTPs means in one tube it may contain one DD ATP, in other tubes contain DD TTP, and in another tube contain DD CTP, and another tube contain DDGTP. Okay, and the DNA polymerase start to add DNTP to the chain until it's happened to add a dideoxynucleotide instead of a normal. Okay, uh, when we kept this reaction tubes, okay, when we kept the reaction tube for reaction, that time this polymerase will add DNTP into the chain. Okay, this polymerase will add continuously add DNTPs into the chain until it's add a dideoxynucleotide instead of a normal one. Okay, until it's add a dideoxynucleotide instead of a normal one. At that point, normal, okay, at that point no further nucleotide can be added, so that strand will be end with a dideoxynucleotide. For example, you can see here these are template DNA. Okay, these are template DNA, and these are primer. Okay, this primer will be marked labeled, and these are template DNA. And here you can see primer that attach, first it's attached to one nucleotide, nucleotide, that's what the polymerase added nucleotide, that is a dideoxynucleotide, that is a dideoxynucleotide. So that growth of the chain is terminated, the growth of that chain is terminated. And here you can see that in this chain you can see added first one guanine nucleotide was, yeah, this likely this guanine was a normal one. Okay, this gone was a normal one, so this allow to add the next nucleotide. Okay, allow to add next nucleotide, but this is a nucleotide, but this a is a dideoxy, but that's why is cannot able to add next nucleotide to this one, so this chain also terminated. Here you can see add a normal sequence here, and is the second one also a is a normal, but c was abnormal. There's a dideoxynucleotide, so the growth of the chain also terminated. Here also you can see, here you see this is a primer, and here also added one guanine normal and adenine normal, and C is a normal, T is normal, G is normal, A is normal, and A is normal, G is normal, C is normal, but T is a dideoxy, so the termination is occur here. So when we are doing this reaction, okay, for tubes we'll do the reaction, we'll get a fragments with a varying length. Okay, we'll get a fragment with a varying length, we'll get a fragment with a varying length, and that DNA, that different length DNA separated by polymerase polyacrylamide gel electrophoresis, and the band will be identified by based on autoradiography. Okay, that means we'll separate this different fragment of DNA using polyacrylamide gel electrophoresis, and will expose this gel for a autoradiography, because that primer is labeled with the radial labeled. So we'll expose this gel for autoradiography, and based on the bands in autoradiography we can construct the sequence. Okay, based on the bands in the autoradiography, that we can construct the sequence. Okay, that means we can identify the sequence based on the bands in the autoradiography. We can see here this figure explain how we'll do this Sanger's method of sequence. Okay, we can see how it will do the Sanger's method of sequence. Okay, here you can see we are taken template DNA primer plus polymerase. Okay, we are taken uh, template DNA primer polymerase DNA polymerase and this normal DATP, normal CTP, DGTP and DTP, and make a mixer of reaction. Okay, make a mixer of reaction. Okay, make a mixture of reaction with a template DNA primer will be labeled. You have to remember that primer should be labeled, and the DNA polymerase, the deoxyadenosine triphosphate, DCTP, DGTP, and the TTP. Okay, and this is a reaction mixture. From that we have to divide this mixture into 4 tubes. Okay, we have to divide this mixture into 4 tubes, and in that tube, the first tube will add DDATP. Okay, DDATP will add DDATP. Okay, DDATP means dideoxyadenosine triphosphate, the first tube, and the second tube will add dideoxy CTP. Okay, dideoxy CTP, and we'll add DDGTP. Okay, DDGTP, and the fourth tube will add DDTTP. Okay, what will add DDTTP. Okay, you have to remember that when you are adding DDNTPs, that means dideoxynucleotide phosphate, that concentration should be less compared to our normal DTPs. Okay, that means the concentration of that DDNTP should be less compared to our normal DNTPs. Okay, normal to normal DNTPs. Okay, that means this DDNP concentration should be less compared to our normal DNTPs. And after that, we'll keep this 4 tubes for reaction. Okay, we'll keep this 4 tubes for reaction. In that case, let's go for amplification. Okay, let's go for amplification, and that amplification vary in length. Okay, that amplification will create DNA with varying length, because in the previous slide we are told that when at the time of amplification, the polymerase will add nucleotides. Okay, deoxynucleotides into the chain. Okay, deoxynucleotide into the chain. When a dideoxynucleotide is added into the chain, that time that chain growth will be stopped. Okay, that polymerase will add nucleotides into the primer, here for example, here you see this is the template DNA and this is the primer. This is the primer. Okay, and here you see that this ATP, then CTP and GTP and TTP, and this also separate this reaction will be occur. The same template DNA will be amplified. Here the amplification will be terminated by DDATP, and this tube the termination will be this amplification will be terminated by DDCTP. Here that amplification will be terminated by DDGTP, and this tube this amplification will be terminated by DDTTP. Okay, when adding nucleotides, the polymerase will add nucleotides into this primer. Okay, into this primer. That time, sometime randomly, this polymerase will add this DDNTPs. That means dideoxynucleotides into this primer. Okay, that time that termination will be occur. Here you see this is the primer. This is labeled the primer. Here in G tube. First that polymerase added DDGTP. Okay, DDGTP. So that growth of this chain is stopped. So okay, that growth of this chain is terminated. Okay, but in the case of A tube, that's why here you see that growth occur and first added when one normal GTP. Okay, normal guanine is added into guanosine triphosphate is added, and after that in that A tube polymerase added one adenine, but that adenine was dideoxyadenine. So that chain is stopped here, and like that, this all the case also is there. Here also you can see one C is the C. Here see that first in the C tube. Okay, C tube first is added guanine and adenine as a C normal one, D normal one, G also normal one, and A also normal one. But and G this G also normal. But here C random the C was dideoxy. So that termination is occurred, and this adding of this dideoxy is the random, so that we'll get DNA fragment with varying length in different tubes. Okay, we'll get DNA fragment with varying length inside. Told that in this tube, that's a DD-ATP will terminate the chain. Here the DD-CTP will terminate the chain, and here DD-GTP will terminate the chain, and DD-TTP will terminate the chain. And after the reaction, okay, we will get varying length of this DNA in this four tubes because of the chain termination of the method of DD chain termination due to the DD-ATP. That's why we will get varying length of DNA, and we'll find out the length of DNA in the piece in this reaction mixture. For that, we'll load this 4 samples in separate wells in a polyacrylamide gel electrophoresis. Okay, and we'll do this for polyacrylamide gel electrophoresis, and we'll find out which well having the first band. Okay, which well having first band. Here you see from here to we get a small fragment will be here and larger fragment will be here, because in the electrophoresis this side will get a small fragment. Okay, that's why, because the small fragment will move faster. Okay, and here you see in G tube, after the running the this gel, and we'll expose this gel for autoradiography, and in that autoradiography film we will find the bands here, and in this G tube we found one band here. So that tube, that sequence G, and because in this tube this band, this strand, small strand, that fragment is terminated by G, the DD and TPG. Okay, so G is there. Here you can see the same corresponding strand. You can see here this is the primer, and here you can see G is attached to the primer, so that band, this chain is stopped. And next of this one we have see here this A in the A tube. You can see so here this sequence is A. So you see here that's when is A come here, you can first is add the normal G and is added A. Okay, so A was dideoxy, so it's terminated. In this case you see here this is the C tube. Okay, on the C tube this came band is in the C tube, so this DNA is this strand and nucleotide is a C, because C is the, so here you see that first a G normal G is added, but in the A this, and second normal A is added, and but C was a dideoxy, because dideoxy, so that here sequence was C, so that's why C is added to here. And here you see the T well you see in the band, so here see the T is terminated this chain, and here see G in the and this well of G you see here, and here this fragment is terminated by G, DDGP. Here is terminated by A, and here is terminated by G, and here is terminated G, and here is terminated by C, and here what is it? Terminated by T. So from this length of different fragment length, we can identify what are the sequence that's present in our DNA. What are the present in the DNA? So here you can see G. A, C, T, G, A, G, C, T. This is the sequence. Okay, this is the sequence, or this is our sequence. So this is the complementary style. Okay, this is a complementary style. From this we have to find out what is the original sequence of our template, because this is the sequence of template. For that we have to convert into this is a complementary style. Okay, this is our original template style. This is a template style. So when we'll get when this will run this gen and autobiography will identify the sequence present here G A C T G A G C T. So its complementary will be the our template. This DNA sequence complementary or the or given DNA template or given DNA template. Yeah. G A G A C T like that. This is the Sanger's method of sequencing. Okay, this is the Sanger's method of sequencing. I hope you understood this. Okay. So I hope you understood this DNA sequencing. Thank you. Thank you for listening.